Hello, everybody. My name is Lydia Poplinski, and I'm doing my research article, a presentation on the recent pandemic virus, SARS-CoV-2. More specifically, the paper looks at the ACE2 binding of dalbavancin and the effective inhibition of this antiviral protein. At the time that this paper was written, there wasn't a vaccine released, so the major goal of this paper was to continue the important research of finding that vaccine for the global pandemic, which has just caused tons of loss of lives all over. So the virus is non-segmented, it's single-stranded RNA, it has these really important proteins on the outside, which are called spike proteins, and they are used in binding to the cells within the body to ultimately transmit the disease. So viruses inject their DNA into, or RNA in this case, into a cell and kind of use that cell for its own purpose. And in our case, it makes us get sick for some. So the spike proteins bind to this receptor called ACE2 and then inject their DNA. So right here in B is the ACE2 sort of model, and then this spike glycoprotein. Lastly, the similarities between SARS-CoV-2 and this protein called SARS-CoV. This virus has an 82% sequence resemblance to SARS-CoV-2, and it also has similar receptor binding domains. And this ultimately hints that you can infer the general mechanism of SARS-CoV-2 from SARS-CoV. So this research paper wanted to look into dalbavancin, which is a potential antiviral agent that can be used to prohibit SARS-CoV-2 from ultimately causing death in individuals by competing for the binding site, which is competing for ACE2 protein binding. So they hypothesized that dalbavancin would successfully do just that. There's not a lot of time in this presentation, so I won't be able to explain many of the methods used in great detail, but I wanted to make sure to mention a couple that are important to the paper. The first one is qRT-PCR. So PCR is a mechanism used for replicating DNA, and this RT just stands for reverse transcription. And this is used to, like I said, amplify DNA and then study that DNA, look at different sequences within it. The ELISA is a very important method or assay used as well. So you can see the general mechanism here on this slide. You start with an immobilized antigen and you add an antibody, a second antibody, antibody, and this ultimately can help you detect this communication and this binding between the antibody and the antigen. In our case, we're interested in SARS-CoV-2 binding to ACE2, and then also dalbavancin binding to ACE2. So this is a really important method that you'll see applied throughout the presentation, throughout the paper. The last method is the Western blotting, and we've done this in lab. It's basically just using electrophoresis to identify the protein as well as the quantity of that protein, and this is helpful in identifying that antigen-antibody relationship as well. The first step for this research is to identify that dalbavancin is the most effective antiviral agent to use in vivo studies. So the first step is let's look at the FDA-approved peptide drug library. There was a total of 10 candidates that could possibly compete for the ACE2 binding site. And so co-immunoprecipitation and enzyme-linked immunosorbent assays, the ELISA that we just talked about, were used to confirm that 3 candidates were the best candidates for binding to ACE2 and ultimately competing for that binding site to prohibit SARS-CoV-2 from binding there. So here we can see that This is a picture of ACE2. This is the binding site right here. And then you have the residues that are important to binding the dalbavancin into ACE2. In B, we sort of look a lot closer into the structure of ACE2. And you can see multiple different arrows and even different amino acids, also different interactions between all of the ACE2 and the dalbavancin interactions, like hydrophobic domains and London dispersion forces and things like that. So the red and blue arrows are where those hydrophobic domains are. And that's seen right over here. You can see that. So this is just the general sort of structure of ACE2, the binding site, kind of begins to show you the idea that these residues are important, and has this general concept of the look, I guess, of this ACE2 important binding site. Ultimately, from this analysis, they concluded that dalbavancin shows strong inhibitory ability, and there are a couple of peptide drugs that successfully inhibited the interaction between SARS-CoV-2 and ACE2. So the next step is to identify whether dalbavancin successfully inhibits SARS-CoV-2 interaction with ACE2. So in part A, we can see that a Western blot was done. And this was performed and indicated that there was less binding of SARS-CoV-2 with ACE2 in the presence of dalbavancin. And this is indicated by the light color right here. It shows that there was potentially just less binding allowed because dalbavancin was present. And this is the lightest color on the Western blot. The rest are a lot darker, indicating that dalbavancin is a really good competitor for binding, but also we have a couple other that are as well. The oxyglutathione and the polymyxin B are also potential candidates for competing with SARS-CoV-2 for this ACE2 binding site. Down here is just a positive control, and we don't have a ton of time in this conversation in this presentation to go through the details of every single figure. So unfortunately, we won't be able to do that. But B was a similar assay using ELISA instead of co-immunoprecipitation, which was found over here in this Western blot. And then lastly, C shows that as they increased the SARS-CoV-2, there was greater binding. This proved that ACE2 and SARS-CoV-2 binded. And so this was just showing, hey, as we add more SARS-CoV-2, you have more binding to this ACE2. Also similar with dalbavancin, it also binds to ACE2. So the SARS and the dalbavancin aren't in the same, they're not competing within these graphs to bind to ACE2. They're just simply proving that this dissociation number shows that dalbavancin binds even better than SARS-CoV-2. The next question they wanted to address was how exactly does dalbavancin bind, and how is it a competitive inhibitor? So graphs A and B show that dalbavancin had low cytotoxicity. That's found in this flat line right over here. It also shows a general increase in inhibition as the concentration of dalbavancin was increased, and so less SARS-CoV was able to bind to that site. So in the experiment was done in Vero E6 cells and CACO2 cells, and that's the difference between A and B, but they both show very similar trends. A similar assay was done in graph C with HEC293 HAC cells. So letter D is kind of analyzing when dalbavancin is most effective, if it's introduced before SARS-CoV-2 is within a certain area, or whether it can be introduced after and ultimately inhibit. And what happened is they figured out that obviously full time when the dalbavancin is introduced, it's able to inhibit the best instead of introducing it just in the very time that SARS-CoV-2 is potentially given. So lastly, in F, we look at the different genetic modifications of the amino acids for the ACE2 binding site. And we look at how that affects the dalbavancin ability to bind. So you can imagine if you get rid of a really important amino acid for dalbavancin to bind, it's going to have a very low amount remaining. And so you're going to have a lower bar. And that indicates that it's really important to have this amino acid instead of this amino acid. And so that really helped clear up that 2 through 6 and 10 and 11 were really important amino acids in that binding site. And so this paper is really trying to understand further what is important and how does this SARS-CoV-2 really bind, and how does dalbavancin prohibit that binding. So now they take their research and they apply it to some in vivo studies. They feel pretty confident now in the dalbavancin's ability to compete for that active site. And so they take it into mouse models. This is really exciting. And right here is where you see the actual working of dalbavancin, that there's less of a viral load when dalbavancin is present, meaning SARS-CoV-2 is just less of a competitor than dalbavancin for that ACE2 active site. And this is also reiterated in B, where we have a group that doesn't have dalbavancin. And you can see all of this pink in here, and what that's showing is those lymphocytes and macrophages, which get sent when there's an infection or when there's a disease that needs to be treated. And so you can see over here, dalbavancin has a lot less of these ultimately lower interstitial pneumonia sort of areas, indicating that dalbavancin is prohibiting SARS-CoV-2 from ultimately hurting us. Lastly, they wanted to do some studies using a monkey model. So a variety of tests were performed to see the overall effectiveness of dalbavancin within just this new group, this new organism. And so they use several different assays, radiographs, administration techniques, x-rays, and right here it shows that there's one group of monkeys that has dalbavancin introduced, and then there's another group that doesn't. And they take different swabs, different blood and serum tests throughout this 7 days of being first infected with COVID, and then eventually the monkeys are sacrificed, and we look at these lungs. I love this term, sacrifice. I think that's so weird that they use that word. But yes, so if we look at B, we can see that the amount of dalbavancin remained relatively constant once it was injected at 0 and 5 and 3. There isn't too much of a lift unless there's an injection. And this really just shows a long half-life. This is really important when you're looking at vaccines. C shows that amongst saline, there were just many pulmonary infiltrations and signs of pneumonia, and just even more of these macrophages you can see in these pink colors. But with dalbavancin, that damage in the lung is a lot less. So this is really promising results and shows that dalbavancin would just be a really great antiviral agent to stop the infection of SARS-CoV-2. So in conclusion, this research article helped us learn about just the basic structure of SARS-CoV-2, ACE2, how it binds, some of the really important amino acids a part of the virus, as well as just the effectivity of dalbavancin preventing entry of SARS-CoV-2, and ultimately kind of indicating that dalbavancin is a very promising antiviral candidate. Thank you so much for listening.